So our next speaker is Raquel Quatrini from Universidad San Sebastián, and she will be talking about acidic microbiomes as model systems in eco-evolutionary studies. You are muted, Raquel.
So can you see my presentation?
Yes, perfect, perfect.
So just a second. I first want to start by thanking the organization for giving me a chance to present our results. I will be showing a little bit of the work we have been doing on acidophilic microbiomes in a wonderful natural laboratory in the north of Patagonia. So with that, I move along. I want to give you just a little bit of frame. We have heard some of this in the earlier talks, but Chile is probably best known to the world by some of its iconic landmarks. The Andes mountain range is one of these, which spans its whole geography, and also its numerous active volcanoes. Yet also for being one of the countries with the largest known metal reserves, particularly copper. Now we hear more about the lithium. Chile is also a land of mining, and both features, the Andes Mountains, the volcanoes, and the mining activities are related to the intrinsic tectonic activity of the region, which spans part of the Ring of Fire. So with that, I want to let you know also that as mineral reserves decline and mineral waste deposits increase, and the mining industry is compelled to adhere to more stringent environmental laws, greener mineral processing technologies are on demand, and this is where acidophiles step up. So microbial extremophiles thriving in acidic environments are natural miners. They are able to produce mineral leaching agents, basically protons and ferric iron, as byproducts of their metabolism, and thanks to these agents, they dissolve the matrix of sulfidic minerals with producing really much lower environmental footprints than conventional mining practices. So this biotech, sorry, my mouse is very sensitive. So this biotech is known as bioleaching, and acidophiles are really key to this practice. So besides occurring in industrial habitats, acidophiles all across the tree of life have been isolated from natural environments. Most of them mineral-bearing environments such as underground sulfidic caves or geothermally influenced sites. We know from the work of several renowned microbiologists working on acidic mine drainage a bit about the ecology of these particular microbiomes. We know that their communities are fairly simple, encompassing a handful of different species, most frequently being dominated by a single taxon, and concurring, depending on the particular habitat types, with a handful of different other acidophiles. So this gives rise to several different possible community assembly variants, whose rules of assembly are more affordable, but which we still do not fully understand. So my lab is actually engaged in trying to figure these rules out, along with the influence and relevance of microdiversity in the systems. As I mentioned, these systems have low complexity, but they seem to have increasing levels of microdiversity. So with this, we pretend, or we aim to generate fundamental knowledge on ecosystem function, but also to provide rational basis for handling and intervention of these communities for the mining industry and the environmental mitigation practices. We are currently using a local natural laboratory, which is shown in the slide. I lost my pointer, sorry. And I don't see it again. Yeah. So a natural, we are using a local natural laboratory to study these acidic microbiomes and to study the population biology of several relevant model acidophiles. This system is the Cabiahue-Copahue volcanic complex, which is located in the Andes, in the southern volcanic region of Chile, in the boundary between Chile and Argentina, in one of the most seismic regions of the world. You can see here in the Google Earth image a view of the caldera. So key to these studies are collaborations in Argentina, UK, Mexico, and Chile, with whom we are modeling, sampling, processing, and modeling the data emerging from the system. But I wanted to mention also that as relevant are the local communities at these locations, which actually interact and safeguard the natural manifestations in the system, and yet are growingly aware of what understanding nature and science can bring, and who have kindly granted us access for sampling and fieldwork. So the main features that have drawn our attention to the system, apart from the volcano itself, are two acidic springs that are located near the volcano crater, which feed with hydrothermal fluids, a glacial watershed that flows down a hill through a series of cascades. You can see, or waterfalls, sorry, that you can see here in the image, to give rise to a glacial carved horseshoe-shaped lake, which drains into the steppe, the Patagonian steppe. So one remarkable feature of this system is the gradient that is established in both pH, temperature, and redox conditions, which give rise to a number of different habitats that are potential habitats for the growth of acidophiles. So we have been sampling the system through both cultivation-based and cultivation-independent approaches, building a fairly decent collection of strains, and now trying to rear in the lab some of the local consortia to do in situ studies. And we have also generated a fair amount of sequence data and environmental metadata to dissect and model these communities. So for the moment, we have performed 4 sampling campaigns of increasing complexity, collected basically water column samples all across the system, spanning a number of local sites and physicochemical habitats. So far, we have analyzed 40 metagenomes. The analysis of this data have shown that bacteria outnumber other types of organisms in the system, with fairly over 90% of the total breed abundance pertaining to bacteria. As a whole, the system harbors a quite diverse microbial community for an extreme system, with different types of organisms and different ecophysiological lifestyles, and covering from extreme acidophiles to non-acidophilic taxa. From these metagenomes, we have so far been analyzing the metagenome assembled genomes, the MAGs, recovered from the systems. Over 2,000 of them are high quality, medium quality MAGs. Most of them have been assigned to classes using genome taxonomy database resources. However, as reflected by this quite simple graph, we see increasing amounts of novelty as we increase the resolution of the taxonomy lens. So there is a quite extent of biological novelty. To reflect this novelty, we have plotted the percentage of MAGs that fail to match known genera and species used as reference for taxonomic classification, and group them by phylum. And in blue, you see this percentage, the novelty. And with few exceptions, most phyla present in the system harbor novelty to a varying extents, from few representatives to the entire bunch present in the system. To illustrate the point a bit further, in the next few slides, I will focus on one particular group we have been studying in much greater detail in the lab, and for which we have recently published a thorough taxonomic revision at the level of class. These are the Aciditiobacillia class members, or Aciditiobacilli, which encompass, after this work, 5 genera and up to 18 species. Most of the species, or many of the species in this group, are relevant model bacterial biomining and bioleaching. And just to mention, the most primitive genera in the class are neutrophilic and acidotolerant mesothermophiles, and the most derived species are mesophilic cyclotolerant extreme acidophiles, which are able to, all the species, to oxidize sulfur, and some of them also to oxidize iron. This group is well represented in the system. It actually encompasses 40% of the total breed abundance in the metagenomes we have produced so far. And they span a variety of sites and habitats up to the lake, where the pH increases above the threshold of established for acidophiles, which is 3.5, and the energy sources begin to decline. So they occur along these sites sorry, I'm moving fast. They occur along these sites in different proportions or total abundance, shown in gray in the background, and also in different species combinations, so reflecting the inherently different community structures of the system along the gradient. We have inspected the MAGs, again, high quality, medium quality MAGs assigned to the class, recovered from each of these sites. And in addition to many MAGs matching the known species, we have a number of MAGs that show signs of novelty. They are MAGs that don't surpass the species threshold cutoff of the best matching species, so clearly showing that there is additional novelty. And the point I want to make by showing this is that novelty is not only in the other poorly studied phyla encompassing acidophiles, but also in the best studied groups of acidophiles that we know about. So we have made some efforts to validate some of these novel taxa. Here I'm showing the results that we have on a genus we have named Igneaciditiobacillus for its origin. And we have looked back out from the system to get a little bit further into this. So combining marker-based analysis, genomic indexes, and phylogenetic approaches, we have been able to expand the genome to a total of 7 species, validating the existence of the genus per se, and also showing that there are differentiation processes to be further studied. Most of the species remain uncultured. Some are actually misassigned strains that have been sitting in the labs for a number of years, unnoticed. And one relevant feature that we uncovered by mapping the 16S data and oligotypes matching these 7 species is that they all map two locations that overlap the distribution of hydrogeothermal terrestrial environment. So showing a very distinctive biogeographical pattern. Knowing this, knowing their preferred niche or habitat, we have emulated the conditions in the lab. So in addition to acidity and reduced sulfur compounds, which these organisms as a class use to grow, we included high osmotic stress, which characterize the sites from where we have isolated these organisms, provided in this case by chloride salts. And we have been able to grow them in the lab and to test their salt tolerance. This is a feature that, move again. This is a feature that is relevant for biomining practices, particularly for the bioleaching of mineral, recalcitrant minerals, which require this combination of harsh conditions. So with that, I would like to wrap up now. Yeah. So I have shown you in a glance about microbiomes inhabiting natural acidic volcanic landscapes and the biological novelty they harbor. Hoping with this to contribute a little bit to the global knowledge on microbiomes from extreme environments, which are only partially characterized, and particularly to increase the representativity of the data in the databases that is coming from South America. Hopefully soon we will have more insight into the assembly rules deriving from this incredibly informative and metadata-rich gradient system. I also wanted to mention that working in this corner of microbiology, we face a number of challenges ranging from sample, which is quite dangerous, to storing of extremophiles, which for instance can't be cryopreserved as most organisms. So we have to keep them as livestock and reculture them periodically. So there are a couple of challenges to help preserve the microbial diversity when we are dealing with this kind of systems. So with that, I would like to thank my lab and institution, shown here in the slide, and our great collaborators, in particular those voted, and our funding sources and authorities in Neuquén, Argentina, that have allowed us access to sampling. And all of you for your attention.
Thank you, Raquel.